Thank you, Lan, for organizing this symposium. I wanted to tell you a true story in this talk. So my lab, as Ning said, works on the ubiquitin proteasome system. We work on kaolin ring ligases, so I don't have to do this introduction since Michele, Ning, and Havram did a great introduction of this system. And in the past year, we've been working more in the space of drug discovery. We have been collaborating with SEED. So I would like to show you one story about development of product to target KRAS G12D. This is a project in collaboration with SEED. And then I'll show you some data that we recently sent out for publication on a method that we developed that is called DBP. It's a method for unbiased identification of protein interacting to drugs in cells. Most likely I don't have to introduce RAS to this audience. RAS is the holy grail of cancer. Most of cancer feature KRAS mutation, especially the deadly one, like pancreas cancer, lung cancer, colon cancer. RAS, it's one of the main upstream effector of the mitogenic signaling cascade, activates the RAF, MEK, ERK pathway, as well as the PI3K pathway and the NF-κB pathway. And there's been an attempt to target, of course, RAS for therapeutic purposes in several ways. But now, with the protein degradation, we can actually try to target RAS at the degradation level. As many of you know, RAS is a GTP binding protein. So in normal cells, RAS can cycle in a GTP to a GTP bound state, and this happens to the help of GAP that can exchange the GTP into GDP, and then GAF that exchange the GDP into GDP. And so this cycle turns on and off RAS and makes sure that the system is a homeostasis. However, in cancer cells, oftentimes we do see mutation of RAS in position G12 or G13. And if you look at the three-dimensional structure of RAS, you can notice that this is the GTP pocket of RAS, and here are switch 2, switch 1, and P-loop. And now mutation in the G12 or G13 that are in the P-loop side, they lock RAS into a GTP-bound state, hence having KRAS constitutively active. Now, 2 years ago, Mirati developed this molecule called MRTX1133, which is a selective KRAS G12D inhibitor. So it's a pretty complex molecule, I'm not a chemist. So the way I understand is that this molecule fits in the switch 2 of RAS and then extend into the GTP pocket of RAS, locking RAS in a GTP-bound state and dissociating RAS from RAF and as such inhibiting RAS signaling. The molecule is pretty good. Binding data are in the nanomolar. And also if you look at toxicity in cells, these are in cells that are KRAS G12D mutated. So we're seeing on the IC50 about 6 nanomolar, while the IC50 in KRAS wild-type cells is about exceed the 3 micromolar. So in our lab, we developed this cell line called, this is a pancreatic cancer cell line called ASPC1 that has endogenous target of KRAS G12D with this short peptide high beat. High beat, probably many of you knows, is a little peptide that binds the large beat protein and recreate the nanoluciferase. And so we generated this line. Importantly, ASPC1, they have a biallelic mutation of KRAS G12D. So all the pool of RAS that we do see by luminescence or by Western blot is actually mutated RAS. There is no wild-type RAS in this cell line. So in our lab, we made a clonal selection of these cells that we generated by re-expressing large beat. So now high beat KRAS can bind large beat that can give us a luminescent signal. And we know that that's RAS because if we knock down RAS, the signal, the luminescent signal go down. We cannot CRISPR out KRAS because these cells are completely dependent on KRAS. So we use these cells to screen a selected library of compound that came from seed. So seed is interested in develop KEAP1 based product. So the KEAP1 ligand came from a paper published about a year ago from the group of Nathaniel Gray. And they use this ligand to attach it to the MRTX113 with several types of linker. And so we screen a mini library of this compound. And we noticed that only these two compounds could actually downregulate a little bit KRAS. And actually, these two drugs that we call it A44 and 719. So the A44 is the pro-drug that gets metabolized into 790. So effectively, they are the same compound. So these two drugs had a little bit of effect. And then we had a VHL version of this KRAS G12D product as well. So we validated this by Western blot. And so by doing fractionation, because this cell line, they only express KRAS G12D, so the whole pool of KRAS is actually membrane bound, as you can see in control. When we treat with the KEAP1 product, the 790, we do see a slight downregulation of KRAS. Well, instead, the VHL product seems to be much more effective in downregulating KRAS. And this was also confirmed in MLN 4924 rescue experiment. As Michele told you before, MLN 4924 is an inhibitor of kallin deletion, so we can block all the kallin ring ubiquitin ligases. Here, if you treat with the KEAP1 product, the 790 or the 844, you get very tiny degradation of KRAS. The VHL product instead is much more potent and can be rescued by MLN 4924. Importantly, if you use this compound in a cell line that express wild-type RAS, there is no effect, suggesting that these compounds are actually active on the G12D compound. So we were quite disappointed from the result that we get with the KEAP1 product. And so until we did this experiment, and I'm not sure why we did it, but anyway, we did it, and we got this result that turned out to be quite interesting. So here is a dose-dependent increase treatment of ASPC1 cell line with KEAP1 product, and literally we do not see any effect on KRAS G12D level. This is the VHL product. At 1 micromolar, we do see a pretty good degradation, and then we start to see the hook effect. So I haven't seen anybody talking about hook effect, but this is something that with product happen quite a bit, and it's a problem because while in cell line you can titrate the concentration of your product to make sure that you are outside the hook effect, in vivo you have no idea of this, and so you just treat mice or eventually patients with the product and hope that your concentration is below the hook effect. Anyway, we're starting to see this hook effect of 2.55 micromolar. However, when we mix the VHL and KEAP1 product, not only we see a synergy at 1 micromolar, but then we also see somehow a release of the hook effect that we see with the VHL product only. And we wanted to be a little bit more sure about this experiment. So we did this other experiment in which, so just follow me. So this is DMSO. So this is the level of KRAS G12D. Now this is VHL product used at 1 micromolar. We do see pretty good degradation of RAS. Now we use it at 5 micromolar and we generate the hook effect. So there's no degradation of RAS. Now on top of the 5 micromolar concentration of the VHL product, now we add the KEAP1 product. And this is a time kinetic 5, 8, 16 hour. And we see that KRAS protein now gradually goes down. So somehow the hook effect of a VHL product can be released by the concomitant use of a KEAP1-based product. And this is true also in washed out experiments. So in here, again, we generate the hook effect using 5 micromolar VHL product. Then we wash out the VHL product, and then we just add the KEAP1 product. And again, we do see this downregulation of KRAS, almost suggesting that the VHL product leaves some sort of remnants on RAS that then is taken upon by the keep one product that is able to continue the job. So to see whether there was a synergy also in the ubiquitination of the target, again with SID we developed this in vitro assay using cell lysate. And Avram has a little bit talked about lysates as a way to use for ubiquitin reaction. So we took our ASPC cell line, we did the lysate preparation. This is quite complex procedure. Happy to talk to you if you're interested. And then in lysate, so this is all with endogenous component, we add the products, and then we add a ubiquitin mix consisting of E1, a couple of E2s, some DAB inhibitor, flag ubiquitin, and a system to regenerate ATP. We let the reaction go, then we denature those extracts, we boil them, we dilute them, we do flag IP for anything that is being ubiquitinated, and then Western blot analysis. And so this was the result. So when we treat these lysates with these lysates with the keep one product at 0, 10, 100 micromolar, nothing really happened. So we don't see any ubiquitination of KRAS. When we treat with the VHL product, we start to see some ubiquitination at 10 micromolar, a little bit stronger at 100 micromolar. However, when we mix these two, the keep one and the VHL, at 10 micromolar, we see a much stronger ubiquitination. So compare this versus this versus this, which suggests that the two products are synergizing in ubiquitinating the target. We made sure that this ubiquitination is keep one and VHL dependent. We create a number of keep one knockout cell line using CRISPR technology. Same. We created VHL knockouts, and then we repeated the experiment. The combination give us ubiquitination. However, if you knock out keep one or if you knock out VHL, then you lose this ubiquitination. We're starting now to a little bit analyzing the effect of this molecule in vivo. And just as an example, this is a colony formation, and this is the treatment of the keep one product. This is the VHL product, and then we start the combination. We do see a synergistic effect, and this is a quantification of the experiment, representative experiment shown here. And next we ask, is this generalizable? Is this true only for this KRAS product, or can be true also for other targets? So we turned out into AR, so degradation of AR. So again, SID synthesized for us a keep one based version of an AR product, and this keep one version had literally no effect on the AR level. The ARCC4 is the VHL version for AR product, and here we see a decent degradation, I would say, at a DC50 in the low 500 nanomolar. And this is the Arvinas compound that is a cereblon-based compound that is much more potent in inducing AR degradation. Already 100 nanomolar, you see almost full degradation of AR. And so again, now we try to mix the two products and see the effect. And so again, follow me. So this is the MSO treatment. This is 1 micromolar, keep one product, no degradation, 0.1 micromolar, VHL product, again, very tiny degradation. When we mix these two, now we get degradation. So this 3 is the control, and this is the mixed effect. And here is a quantification of this data. So this is where we've gone so far. There's still a lot that we don't understand. But a possible model that we're going after is the following. So when we have a target and we use a product that is VHL-based, now some pool of the target gets fully ubiquitinated. This target now is degraded. There's no problem. However, there might be a pool of the target that might not be effectively ubiquitinated. There might be small chain that are being added, or that is the consequence maybe of the hook effect or some low efficiency ubiquitination process. Now this hanging fruit, now they can be taken over by keep one. So if you add the keep one product now, you could probably elongate this chain and then promote an effective degradation. So this is more or less the model we're going after, and I'll be very happy to know your thoughts. So for the first part of the talk, here is a summary. KEAP1-based product are weak degrader KRAS G12Ds as opposed to VHL product. VHL-based product display the classic hook effect on target degradation. KEAP1-based product synergize with the VHL-based product in promoting KRAS G12 degradation. KEAP1-based product release the hook effect of VHL-based product, and also KEAP1, a VHL-based product synergizing in promoting degradation of androgen receptor in prostate cancer cells. More ongoing work that we're doing with SEED is the assessment of KEAP1 and VHL-based product in cell line and in mice, assessment whether KEAP1-based product can release the hook effect of VHL-based product with concern the androgen receptor in prostate cancer cell line, and whether this combination can be also effective in prostate cancers model as well. So with that, I'll switch to the second part of the talk instead, that is more a method-based talk where we develop this TAP-DBP, which stands for Tandem Affinity Purification for Identification of Drug-Binding Protein. And so this project was really inspired by the sad story of thalidomide. You all know thalidomide was used in the 60s as a sedative for pregnant women, and then actually in the US was never marketed, but in Europe made quite a bunch of damage. And now all these kids that were born with phocomelia, they were named actually the son of thalidomide. And then it came back in the 1990s and 2000. It got approved for MDS and for multiple myeloma. Up to 2010, we actually had no idea what's the target of thalidomide until the lab of Handa in Japan did this experiment where they cross-linked thalidomide to beads, and then they used a lysate, ran it through these beads, and they then did identified by mass spec cereblon. And then, as you've seen also in the past presentation, since 2010, fast-forwarding identification of Icaros as the target of thalidomide, and then PROTAC, and here we are. Well, inspired by this story, we were wondering whether we can develop a more unbiased method to identify protein that interacts with drugs in cells. And so we developed this cassette that we call N-turbo FKBP12 cassette. Essentially, this is the cassette. It has a flag HA tag with nanoluciferase, turboid D, and thanks to Lan, I don't have to explain you what is turboid D, and then FKBP12, the mutant that is F36-V. Now, this mutant of FKBP12 can bind a ligand to a very high affinity. And so the idea here is to link whatever drug that you might think of to the FKBP12 ligand. So express this cassette in cells, and we have this cassette in several vectors, including retro and lentivector. So we can stably express this cassette. Now treat your cells with your bifunctional molecule that consists of the FKBP ligand plus the drug of interest. This, the drug target gets recruited close to FKBP12. The turboid D can biotinylate whatever is in proximity of this interaction. And also the flag HA module allow us to do purification in native condition. So first of all, we validated this idea. And to do so, we used the DTAC13 from the Ben Eber lab. So this is a very well-known molecule that has an FKBP12 ligand, linker, and then the lenalidomide, the cereblon ligand on the other side. We treated cells that expressed the FKBP12 cassette with this DTAC13. We saw rapid downregulation of the cassette, meaning that there was degradation going on. Degradation is MLN dependent because it can be rescued with MLN for 924. The nanoluciferase cassette is very useful because it allows us to do this experiment really quickly in a high throughput format. And biochemically, we can IP the N-turbo FKBP cassette in presence of DTAC13, and we can identify interaction with cereblon and DDB1 only when we treat with DTAC13. And of course, we need to treat with MLN to avoid any degradation going on. So at this point, here is the method. So we express the FKBP12 cassette in cells, we treat cells with biotin, and then we perform a first IP in native condition using an anti-flag. And then we do an SDS solution. So we break all the protein-protein interaction. Then we perform a first mass spectrometry in native condition. And then we perform a second purification using streptavidin-coated beads. Now, these beads will catch only those interactions that are in proximity of the drug interacting protein. And we perform a second mass spectrometry experiment to identify the primary target of this compound. And so again, with DTAC13, here is the biochemical experiment. This is the input, this is the IP, this is the native IP. This is the IP of the FKBP12 cassette. It binds cereblon in presence of DTAC13, binds cereblon and DDB1 in presence of DTAC13. And then also in presence of biotin, we do see interaction with the DTAC13. However, when we elute this and we perform a second purification step, now we can only see interaction with cereblon only when we add biotin, and when we add DTAC13, we do not see any interaction with DDB1. When we do this in an unbiased manner, the first IP, we can identify all the cereblon complex, including DDB1, cereblon, Cal4B, and we have other members of the complex here, depending where you set the threshold. The second IP identify really only cereblon, and so if you combine the two data sets together, now you really see only cereblon as the main interactor of DTAC13. So the method worked with DTAC13. What about other drugs? So in collaboration with Joe Salvino at the Wistar Institute, we utilize olaparib. Olaparib, for those of you who don't know, it's a very good inhibitor of PARP1. And so again, this is the FKBP ligand, this is the olaparib. We design two molecules. We call it olaparib 1, olaparib 2. Olaparib 1 has a more flexible linker. Olaparib 2 has a more constrained linker. Again, we first do the biochemistry, input, IP. So this is the IP. When we treat cells with olaparib 1, olaparib 2, we only see interaction with PARP1. The controls are clean, and we use also DTAC13 as a control that binds only cereblon, doesn't bind PARP in first IP, and then in second IP with the biotin, again, we find olaparib 1, olaparib 2, same for cereblon. So biochemically, this makes sense. Does it work also unbiasedly? So here is the mass spectrometry data. First IP, we identify a bunch of interactors of olaparib, in this case olaparib 1, but the same is true for olaparib 2. And I just noted where PARP1 and PARP2 are. And then second IP, streptavidin pull down. Again, we identify PARP1 and PARP2. And when we merge the data set, we basically find that olaparib really binds only PARP1 and PARP2 in cells. And this was actually surprising how clean is actually this drug. We were hoping to find something else. Anyway, this led us to the design of a product for PARP. Here we use a MDM2 ligand, and so this is an MDM2 linker, olaparib, PROTAC, and this is an in-cell Western blot. Interestingly, we do see a very nice degradation of PARP1, a low nanomolar concentration. However, despite olaparib binds PARP2 to the same extent as PARP1, we do not detect any degradation of PARP2. So I'll stop here. This is also a work in progress. I've shown you that this N-turbo FKBP12 cassette allows for efficient binding of the FKBP12 ligand in cells, and N-turbo FKBP12 cassette allows for tandem affinity purification of drug binding protein. And this cassette allows for identification of the primary target of drugs in cells. We're very interested in the future of this technology. Of course, we're working on other PARP inhibitors with the rationale that there are some PARP inhibitors that they bind PARP to the same affinity, but they have a very different cytotoxic effect in cells. So one possibility is these other PARP inhibitors, they have other target. We're using also this method to study drug resistance. So for instance, oftentimes when we use, for instance, Palbo, we have an experience using Palbo, we have noticed that the Palbo start to bind to other targets. And so we're using this method to find target that might explain drug resistance in cancer cells. And we are also interested in partnership with whoever is interested in identify intracellular endogenous target of drugs for which there is no really mechanism of action. And so I'll stop here. I just want to thank Sevanul, who is here in the audience. He led both studies, a great postdoc in the lab. He was helped by an undergrad student, Christopher Hu. And then all the people at SEED. It's been great working with you guys. Present and past, the team has changed a little bit, but it's been a fantastic experience. Advisor, Michele Pagano, Nin Zheng, and Avra Mersko, and also, especially for the second story, the people at WISAR, Joe Salvino and his postdoc Jitendra Gaur, as well as the proteomic people, Hizing Yao and Thomas Beer. Some of the funding for this project, and happy to get any questions.